Hello everyone, I'm Bharat Singh Ilham from Department of Food Engineering and Technology, Tezpur University, MTech second semester. Today, I'm going to give a presentation on the use of freshness indicator in packaging. Here are my contents for the topic of my presentation. Introduction, compounds indicating the quality of packaged food products, freshness indicators, future trends, and reference. The use of freshness indicators in packaging. As we all know that the freshness indicators supervise the quality of food products during storage and transportation. A freshness indicator is used to indicate directly the quality of the product. The indication of microbiological quality is, for example, based on a reaction between the indicator and the metabolites produced during growth of microorganisms in the product. In the figure 1, it shows the use of the freshness indicator, for example, time temperature indicator and leak indicator. They help us to find out the microbiological growth or microbiological contamination, helping them to find out the freshness of that particular product. In this slide, we are going to discuss about the compounds indicating the quality of packaged food products. An essential prerequisite in the development of freshness indicators is knowledge about the quality indicating metabolites. Some of the quality indicating metabolites include glucose, organic acids, ethanol, volatile nitrogen compounds, biogenic amines, carbon dioxide, ATP degradation product, and sulfuric compounds, etc. Let's discuss in detail. Glucose. Glucose is an essential substrate for many spoilage bacteria in air, vacuum packages, and modified atmosphere packages or MEF. As bacterial growth takes place, glucose is depleted from meat surface. The measurement of the glucose gradient could be utilized to predict the remaining shelf life of the particular food product, which was being proposed by Chris Rogers in 1993. Organic acids. Lactic acid and acetic acids are the major compounds having a great role in glucose fermentation by lactic acid bacteria. The amount of L-lactic acid was reported to decrease during storage of fish and meat. However, the concentration of D-lactate was reported to increase during storage of meat, which is a more promising freshness indicator. With modified atmosphere packaging in poultry meat, the concentration of acetic acid in the tissue fluid and homogenized meat was increased, which is a function of storage time and temperature. Ethanol. Ethanol is another major end product of fermentative metabolism of lactic acid bacteria. An increase in ethanol concentration in meat and fish indicates an increase of total viable count of the product. On an average, the concentration of ethanol was increased as a function of storage time in ice fish. Raven in 1993 studied the formation of ethanol in smoked vacuum-packed salmon. The concentration in stored fish samples were considerably higher than in fresh ice fish. At the end of the shelf life, concentration as high as 10 mg per 100 gram fish were observed. Renden and its team in 1995 found that the amount of ethanol in packaged headspace of modified atmosphere packaging in marinated rainbow trout was increased together with storage time and size of the package leakage. The ethanol concentration in the package headspace containing marinated chicken pieces in modified atmosphere, 40% carbon dioxide and 60% nitrogen increased as a function of storage time. Volatile nitrogen compounds. High level of basic volatile nitrosyl compounds like ammonia, dimethylamine, and trimethylamine give an indication about microbiological spoilage of fish. Trimethylamine, formed by microbial action in fish muscle, was a major metabolite responsible for the spoilage odors of seafood. Biogenic amines. Biogenic amines such as triamine, cadaverine, putrescine, histamine are especially widely considered as indicators of hygienic quality of meat products. Due to the health risks, a tolerance level of 100 mg per kg of fish has been established for histamine by FDA, Food and Drug Administration. Putrescine and cadaverine are formed from ornithine and lysine, widely suggested that these diamines are indicators of the initial stage of decomposition of meat products. Carbon dioxide. Carbon dioxide is generally known to be produced during microbial growth. Carbon dioxide is also typically added as a protecting gas to modify atmosphere packages together with inert gas nitrogen, since it has bacteriostatic effects. A modified atmosphere package for non-respiring food typically has a carbon dioxide concentration as high as 20 to 80%. It is possible to use the increase in carbon dioxide concentration as a means of determining microbial contamination in other types of product in MAPs already containing a high concentration of carbon dioxide. ATP degradation products. K-value is defined as the ratio of the sum of hypoxanthine and inosine and the total concentration of ATP-related compounds. K-value correlates with the sensory quality of fish and other types of meats, used as a freshness indicating parameter. For fresh meat, the value is low since the concentration of ATP degradation product is low as compared to the concentration of all the ATP-related compounds. Sulfuric compounds. Some of the sulfuric compounds have a remarkable effect on the sensory quality of meat products due to their typical odor and low odor threshold. Hydrogen sulfide and other sulfuric compounds produced during the spoilage of poultry by Pseudomonas, Alteromonas species and psychotrophic anaerobic Clostridia. Production of hydrogen sulfide can be used as an indication of enterobacteriaceae and hence also of hygienic problems in aerobically stored meat. Hydrogen sulfide production by Enteromonas putrefaction, Enterobacter liquefaciens and Pseudomonas was discovered in high ultimate pH beef from stressed animals. Now let's discuss about the examples of freshness indicators. First, indicators sensitive to pH sense. This is based on the use of pH dye, which sense color in the presence of volatile compounds produced during spoilage. The use of the pH dye bromothymol blue as an indicator for the formation of carbon dioxide by microbial growth has been suggested in many studies. An increase in carbon dioxide can be used to determine microbial contamination in certain types of products. pH dyes reacting to the presence of carbon dioxide used to construct intelligent packaging concepts indicating the ripeness of traditional fermented vegetable food in Korea, which is known as kimchi. Many other reagents or indicators such as xylenon blue, bromostresol purple, bromostresol green, stresol red, phenol red, methyl red, and alizarin among others have been proposed for the same purpose. Besides carbon dioxide, other metabolites like sulfur dioxide, NH4, volatile amines, and organic acid have been proposed to be suitable target molecules for pH-sensitive indicators. Next example is indicators sensitive to volatile nitrogen compounds. A concept reacting to volatile amines with a color sense, hence indicating freshness of seafood, has been proposed by Miller and its team in 1999. The concept consisted of a plastic chip incorporating a reagent containing wick. B. As the label was attached to the package, the sharp barb on the back of the label penetrated the packaging film, thus enabling contact between the package headspace gases and the reagent. As volatile amine passed through the wick, a bright pink color was developed along the wick. Label sensor is manufactured based on methyl red. Red methyl cellulose membrane works on the high in pH due to volatile amines decomposition. It was successfully used for real-time monitoring of fresh meat of boiler chicken. The next example of freshness indicators is indicators sensitive to hydrogen sulfide. Freshness indication is based on the color change of myoglobin by hydrogen sulfide, which is produced in considerable amounts during the aging of packaged poultry during storage. It was found that the color change of the myoglobin-based indicators corresponded with the deterioration of the product quality. The myoglobin-based indicators seem to be promising for the quality control of packaged poultry products. Next example is indicators sensitive to miscellaneous microbial metabolite. Diacetyl is a volatile compound evolving from meat as spoilage takes place, and the dye color could be printed on the surface of a gas-permeable meat package. Diacetyl migrating through the packaging material would react with the dye and induce a color change. Another example of freshness indicator includes pathogen indicators. A system known as Toxin Guard (TM) is able to detect the presence of pathogenic bacteria such as Salmonella, Campylobacter, E. coli 0157, and Listeria with the aid of immobilized antibodies. As the analyte toxin or microorganism is in contact with the material, it will be bound first to a specific labeled antibody and then to a capturing antibody printed as a certain pattern. Another system, which is known as Food Sentinel System (TM), for the detection of specific microorganisms like Salmonella species, Listeria species, and E. coli. This system is also based on immunochemical reaction, the reaction taking place in a barcode. If the particular microorganism is present, the barcode is converted to unreadable. Let's discuss about biosensor. What is biosensors? A biosensor is an analytical device consisting of a biological component specific to the analyte and a physical component which is able to transduce the biological signal to a physical one. In this figure, biosensor use analyte, antibody, protein as an input and transform it into a physical information using transducer. In relation to biosensor, Yano and its team in 1995 developed a tyramine oxidase-based sensor for the quality control of beef. Now let's get some information about electronic nose. By definition, an electronic nose is an analytical tool composed of an array of sensors, for example, metal oxide polymer responding to volatile compounds with changes in their electrical properties. Individual compounds are not separated or identified with the electronic nose. However, the samples can be classified to acceptable or unacceptable according to the fingerprint of the volatile compounds and the results obtained with a reference method. For example, sensory evaluation or microbiological analysis. In this figure, it shows a comparison between a biological nose and electronic nose. Biological nose consists of receptors, olfactory bulbs, and brain. However, electronic nose consists of sensors, preprocessing chambers, database, and pattern recognition system. In electronic nose, there is an interaction between volatile compound and sensors, and sensors help to generate information and transfer to the preprocessing chamber. Further, the preprocessing chamber helps to process those signals into the database system. Finally, pattern recognition system identify the information which has been carried out through the process, and finally identify the volatile compound which is being present in that particular food product by using the electronic nose, and thus helps to identify the freshness of that particular food product. Electronic nose can also act as a promising tool for the quality evaluation of poultry meat. Electronic noses have also been successfully used for the quality evaluation of other products like fresh yellowfin tuna and vacuum packaged beef. Colorimetric nose utilizes the color sense taking place in an area of metalloporphyrin dyes upon ligand binding. The liganding vapors such as alcohols, amines, ethers, phosphines, etc. can be visibly identified. Rep sensors. Rep sense is the first intelligent sensor label that changes color to indicate the ripeness of the fruit. It works through the reaction of the aroma released by the fruit as it ripens. Initially, it is red and then graduates to orange and finally yellow, depending on the selection of the desired level of maturity when it comes to eating the fruit. The figure shows the schematic representation of ripe sense indicator. Let's discuss about future trends of freshness indicator. Today, the commercially available intelligence concepts are labels responding with a visible sense to time and temperature (TTI) or the presence of certain chemical compounds like leak indicators, freshness indicators. It will, however, be very likely that the visible labels will be replaced with more developed types of indicators. The replacement of traditional barcodes by electronic tags in a supply chain is becoming more and more common. In future, the tags will be used not only as an information carriers but also as a miniaturized analytical tools. This quality indicating electronic labels could be introduced, example as chips. These figures are the electronic barcode and electronic chips which can be used to identify the temperature and time for that particular food product to indicate the freshness of that food. The advances in ink technology might enable the use of intelligent printed circuits as well. The advantages of printed structures include a low price and disposability. It is very likely that the advances in electronics and biotechnology, example biosensors, immunodiagnostics, would be followed by emergence of new concepts of intelligent packaging. Profound understanding is needed about the formation of quality indicating compounds, both volatile and non-volatile, and also about their correlation with product quality. On the basis of this information, new intelligent concepts, example reacting to more than one quality indicating compounds, could be developed. Here are the references of my presentation topic on freshness indicators in packaging. I hope that everyone is enjoying the topic, and thank you one and all for seeing my video until the end. Thank you once again.